Now, I would like to introduce you Dr. Mauricio Baptista. He's professor at Biochemistry Department at the University of São Paulo. His researchers are in quantitative measurements in cells, membranes and interfaces, photochemical, photobiology, photodynamic therapy, redox process, and cell death. Thank you, Professor.
I think I don't need this one. Huh? So, thank you very much. For me, it's a great honor to talk to this audience. And I first want to acknowledge, I think this is to acknowledge the people that have organized this, so I put their names here. And I want to mention that I really respect them, not only for organizing this, they are being I consider the bandeirantes of PDT in Brazil. Bandeirantes, people from foreign countries don't know this, but they were the people that come in early development of Brazil, and they went from the coast to the inside of the country, and that's why we have a big country like this. And I hope we have the same with PDT, thanks to these people here in São Carlos. So, it was the first time I put a topic like this, and the reason is because I have two goals on this lecture. One is the main message that I want to pass, is that PDT can be very specific instead of being generalist. And I hope I can explain to you during the lecture what I mean by that. And the other reason, I want to demonstrate techniques, because this is an advanced school. So I want you to know what kind of techniques we use in the lab in order to reach some conclusions about this. So in the lab, we basically work with biophysical and biochemical techniques. We run spectroscopy, surface techniques, microscopy, and chemistry and biochemistry. We use solution experiments. We do mimetics. Professor Mosley mentioned that membranes are very important for the effect of PDT. We completely agree, and we study a lot of processing membranes. We also do cell culture, and we do some tissues. But we stop here. In my lab, we don't do any animal experiments, so very few of them. I have the pleasure to contribute and to participate in the work of some doctors in Brazil. And I really am very enthusiastic about their work. We publish papers together. These papers are being well cited, they are being recognized, and the main thing is to develop low-cost PDT protocols. I was happy to know that Foscan is expensive, even for UK, but for Brazil it's really amazing. It costs $4,000, 4,000 euros, a dose of Foscan here in Brazil. So it's really, you cannot talk about any kind of public health using these kind of drugs. But using very low-cost phenothiazines or hypericin, these doctors, they have done wonderful work. And I want to call your attention for the lecture of Dr. Tardivo tomorrow. He'll mention more details about this. I just want to mention the last work he's working on. I don't know if he's going to mention this tomorrow, but probably he will. Just to tell you that he's running control experiments now to prove that PDT can save the extremities of diabetic patients. So come tomorrow to attend to this lecture. In my case, when I started to get interest in PDT, I was looking into this scheme, and I was thinking, what else can we talk about? What else can we learn about this? If we trust that this is all that happens, so I should choose something else, because this was new in 1968. This mechanism of photooxidation, of biological photooxidations. So basically, as Dr. Mosley mentioned, we have mechanism type 2, that's based in single eloxygen, and mechanism type 1. So if we make more of this, we kill more cells. If we make less, we kill less cells. This is what I call generalist. Is this true? Is this really happening when the treatment is happening? Or is this something else going on? So I want to ask you, if this is the only thing that we should think, there are two things that we should consider in order to develop new drugs. we should make a drug that makes a lot of single oxygen, and that it's really happening and it's taking effect. People are getting better drugs every year. They are more expensive, but they are better. They do less photosensitivity, they are more efficient, and they make more single oxygen. And also they absorb more into the red. And the reason why this is important is because this is, if I get the red here, would penetrate much more. You would see my whole finger red. This doesn't penetrate a lot. So if you go further red, penetration is better in the tissue, you can treat more deep tissues. So the question I made several years ago, is this really the important parameters to maximize efficiency? Should we think only of single oxygen yield? So I want to give you some experiments to say that this is not always the case. So I want to show you data comparing two molecules. One of them, this is methylene blue, is a very efficient photosensitizer, at least in cell culture. You can have problems in tissues if it gets reduced, but it makes 50% of triplets and a lot of single oxygen. Crystal violet is also used in PDT in certain cases, and is a very poor photosensitizer. The yield of triplets much lower than 1%, and it makes a little bit of radical species, but it's still a very poor photosensitizer. So can we compare these in solution, in cell studies? And here are survivor curves in the dark, under illumination, in the dark and under illumination. We can measure incorporation rate, so you know how much dye is inside the cell, therefore you know how much light is being absorbed. And then here we have some kind of PDT efficiency that's corrected by amount of light absorbed. And what we see here is that crystal violet at low concentration, this 1 micromolar, is actually more efficient than this one that's methylene blue. And if you increase the concentration to 20 micromolar, both of them become similar. So why is this happening? Why is this molecule that's a very poor photosensitizer is killing cell more efficiently? And so before I go into further details, I want to go back a little bit into the one of the techniques we use. We measure single oxygen in two ways. Physically, we measure the emission in the near infrared. That's a fingerprint of single oxygen. We measure its emission spectra at 270 nanometers, and we measure its rate decay. So this is a decay in solution, water solution. It's very quick. It's like 4 microseconds. So that's why we think if you generate single oxygen, you have not a lot of diffusion time. You react with some target in tissue. And if you have some species that react with single oxygen, the lifetime reduced. So we can measure reactivity and the amount that's generated. And now we are investing in new equipment that is to couple this to a microscope. And I hope in the near future, I can come to show you some data in which we have in a cell or tissue, where is this being generated and how is the reactivity in certain tissues. So let's then look at this data in a 20 micromolar condition in which both of these cells, they have the same killing ability. And then we measure amount of single oxygen. Methylene blue inside the cell is generating 15 times more single oxygen than crystal violet. So then you can think, oh, perhaps it's then the fact that crystal violet is making some more of the other reactive oxygen species, making some reactivity by type 1. And then we measure other kind of peroxidation products by other methods. This is oxidation of dichlorofluorescein. That's a dye that gets oxidized, and we see how much reactive oxygen species is being generated. And then you see that still methylene blue makes much more oxidation products than crystal violet. So we still have the same question. Why is then these two killing the cell in the same way if this is making much more reactive oxygen species? And so then we went to see localization studies. We put these dyes in contact with the cell for some time, and we put molecules that target specific organelles. So here we put the dye together with rhodamine 1,2,3. That's a molecule that goes to mitochondria. If we use like 200 nanomolar or 100 nanomolar of this molecule, we can see only mitochondrial market. If we put more, we'll see the whole cell green. So be careful. You want to do co-localization studies, you want to do nanomolar concentrations. And we put crystal violet, also in the nanomolar, and you see the red dots, and when we put one of the other, you see all yellow, all orange. It means that this molecule, it goes 100% to mitochondria. It doesn't go to lysosome. We put lysotracker, this molecule that mark lysosome, doesn't go there. We put VOGP that mark Golgi apparatus, doesn't go there. It doesn't go to nuclei. It only go to mitochondria. Now you do the same with methylene blue. When we start doing methylene blue at 300 nanomolars, we see nothing. There is no emission. And we know the reason. The reason is that because methylene blue gets reduced and gets electron and it's bleached. It doesn't emit light. It doesn't make any single oxygen. So at low concentration, that's the reason why methylene blue is not so efficient. And where it's going, this concentration? It's going to mitochondria. Here's the rhodamine blue marker. When we put methylene blue at this concentration, you see that the marker is getting spread all over the cell. It means that methylene blue went there and reduced the mitochondrial potential so that this dye is being released. So it is going to mitochondria, but when it goes to mitochondria, it gets reduced. So it's not as efficient to do damaging mitochondria. And it goes to lysosome at higher concentration. So what we learn with this? We learn two things. This molecule, that's a very poor photochemist. It doesn't do any triplet or any single oxygen, very little amount. It can be more efficient than this one that does a lot. And the main reason is because it goes to this specific place, to mitochondria, and do some damage there. So that's the first thing we want to conclude. And that tells us that we can maximize photodynamic therapy looking for selectivity instead of making more reactive oxygen species in the cell. So we think this is our route. We can take a look in how to make molecules that are designed to be more specific to certain localization. Okay, is there any questions so far? Can we progress? Does this has anything to do with the title? Yes, please. I didn't hear. Wavelength? You mean the time of reduction?
What's the lifetime?
The kinetics of reduction of methylene blue inside the cell?
Yeah.
I don't know that for sure. But it's around tenths of minutes.
Okay.
But we never really measure precisely. The absorption wavelength for crystal violet is 59. And for methylene blue is 60-50. We use different lights to make sure that the amount of light absorbed will be the same.
Then the question is, we have different scattering on the cell structure.
Yeah, but that would favor methylene blue.
That will not...
Yeah, I mean, because it's only one cell, it's a monoculture. So there may be more scattering in the orange light that we use for crystal violet, but still that would disfavor his efficiency anyhow. But methylene blue, we excite in the red, there is less scattering.
Yeah, but scattering helps sometimes because you
Okay.
spread light in
I
many
see.
different places. You don't get
I
shadows.
understand.
You know.
Yeah, but this is not tissue. This
Yeah.
is a cell culture.
Okay.
So in order to get more details on how, which other factors affect the efficiency of a molecule, we have been doing a lot of studies by keeping the center of the molecule the same and varying the side groups. So I want to mention a little bit some of the work we've done. I want to mention the work that has been done with Professor Oliveira and Professor Uchoa. Oliveira is right here. They have been very clever in designing molecules that avoid aggregation. Aggregation is one of the factors that decrease efficiency. And they are able to design things that are avoiding aggregation in very high concentration. So if you have any question concerned with that, talk to Professor Kleber. He's sitting right there. I'm mentioning a little bit about the membrane studies we've done. So is it how we can favor the efficiency of membrane damage? And the answer is very simple, actually. if we have a molecule that's an amphiphilic molecule, it means if it has polar groups in one side of the molecule and apolar groups in the other side, this molecule will probably enter deeper in the membrane, have a higher concentration of oxygen to react, and cause more damage because of that. And we measure in this series that we prepare molecules that have the same groups, but internal group, but the same photosensitizer with different side groups. And this molecule that has the cis position is much more efficient than this other that also has two sides positively charged, but are in symmetric position. And the reason is because this goes deeper in the membrane. So here is how these cells destroy, for instance, the membrane of erythrocytes, the red blood cells. This one that has 4 charges, we have to irradiate for more than 1 hour to have these cells to die. And this one, less than 1 minute. Much more efficient. And this is related with the log of dP, that partition coefficient, but not so much, because if the molecule has a cis configuration, interacts with the membrane in a way that the log dP, the partition octanol water, cannot predict. And we are glad to have this data done in 2012, when we have a group of people that was led by Professor Corneiro in UFABC. And when they calculate this, is this true? We were talking about this 10 years ago, but he wanted to make sure that dynamic simulations would tell that that's the truth. And really what he saw, he put the membrane in the computer, he designed the molecules, and he let the species react with each other, and he was able to predict reactivity very similar to ours. The only one that is out is the transposition. This one, we predict a reactivity larger than he saw in the computer. We don't know exactly why. And, but I think we are pretty sure that this is happening because the molecule enters deeper in the membrane. So can we then, if we have some control of what the molecules can do, can we selectively ask these molecules to perform certain tasks in the cell? And the task I'm talking about, can we control this? Can we say, I want this molecule to kill this cell by necrosis, or I want this molecule to kill this cell by apoptosis, or by autophagy, I want to let this cell go to senescence. Can we control this by light? So that's one of the main goals in the lab in the last 5 years, and I'm going to show you some of the results we have now. So this is, necrosis is, I call this is a cell death that's not controlled. If you read the literature, there is today controlled necrotic cell death. I'm not talking about that. I'm talking about uncontrolled necrotic cell death. This apoptosis and autophagy are the 2 other main mechanisms of programmed cell death. Professor Mosley mentioned this. Is a cell death that is somehow inside of the cell. The cell has already this mechanism to trigger this cell death if something goes wrong or if it needs to disappear. A very clear example, we have 5 fingers, and when we were developed in the fetus, these cells that were here had to disappear, and they die by apoptosis. Autophagy has some other things I'll mention in the future. So let's see, how can we control this? If you read the literature, some other people have already say and showed for sure that PDT is able to induce apoptosis versus necrosis. This is basically done by Professor Kessel in the US, and he was able to show this long time ago. And other people, Jory that will be with you here, Agostini, Scott Cheever, some people have done that. They have shown that at certain conditions, if you treat the cell in this way, the cell will die by apoptosis. And autophagy also, Kessel has done some work showing that if he treats the cell with a molecule that goes in the lysosome, the cell can go to autophagy. Our approach is the following. Can we understand this better by doing structure-active relationships? And how can we use this to improve the efficiency of PDT? That's our 2 questions we want to answer. So let's start with apoptosis. So I want to mention one example we have done, that is we have this molecule that's a porphyrin. very low solubility in water. It doesn't dissolve well. We have to dissolve in the MSO and then put a little bit in water. Here, the solution. When they treat the cells with this molecule, these molecules go to the membrane. Okay, you can see here. And when you shine light, the membrane is damaged, and propydium iodine, that's a dye that goes inside the cell that has the membrane damaged, shines up. So this cell is dying by necrosis. Now, if you put the same molecule in a nanoparticle that's prepared by a conservated capsule method, Menger devised this some time ago, and then we treat the cells with this molecule that has been put inside the nanoparticle, and now what we see is that somehow this molecule jumps the membrane. It doesn't accumulate in the membrane any longer. In the cytosol, it's released. We know that because we mark the capsule in blue, and the molecule has fluorescence in red. You see they're different here. So it's releasing the molecule inside the cell. And when we shine light, we see now apoptosis. And we know that because we have caspase 3 response, we have cytochrome C release, and we have also marker here of the membrane with an antibody that recognizes negatively charged lipids that are exposed in the process of apoptosis. So you see that we can, by using a certain way to deliver the drug, we can control from necrosis to apoptosis. How about autophagy? Can we do something about that? So let me go first to say what is autophagy. So autophagy is a mechanism of cell homeostasis. It basically eats things that are waste inside the cell. So if you have a mitochondria that's damaged, there is a mechanism to engulf this mitochondria, do a lot of hydrolysis, a lot of enzymes destroy the molecules, and this can be used to build new molecules. So this is very important for cell homeostasis. If somehow the autophagy is with problem, there is less autophagy than it should be, there is accelerated aging. The cell cannot keep up with itself. But if there is an excess of autophagy, the cell can start killing a lot of things, eating a lot of things, and then it destroys itself. So the cell death by autophagy. We are usually here in equilibrium with that. So can we use light to somehow control this to go in a certain direction? Can we stimulate autophagy? That's the question. So we did compare here two molecules, one that has a dimethylene methylene blue, this is methylene blue. Okay, very similar molecules. This one a little bit more hydrophobic. Because of that, it binds more to the membrane. If we do any kind of assay, I'll mention a little bit to use which we do. We basically can have membranes in solution. We put these molecules together with the membrane, and we do centrifugation, and you separate what is in the membrane, what is in the supernatant. And then we know how we can do binding constants in this way. And we can also see which of these molecules cause more damage to the membranes. There are several ways we do that. One of the ways, we use a system that we put dye inside here, and this dye is self-quenched. Doesn't emit light because there's so much concentration of it. And when the membrane is ruptured, the emission comes up. We can also do giant vesicle studies. You're not seeing well here. But it's we see in the microscope the membrane getting broke. And that's another thing we do. But let me show you some data, because I'm putting here, the membrane damage for methylene blue is only 3%, and dimethylene blue is 92%. How do we know that? So this is the assay that we can do. With this self-quenched molecule, we put these vesicles here, and you illuminate in the presence of dimethylene blue or methylene blue. And when it's methylene blue, nothing happens. There is no increase of emission. There is no membrane damage. And when this is done with dimethylene blue, there is an emission increase. These are the molecules we study. I'm not mentioning here, just these two I want to compare today. So this is much more efficient to cause membrane damage. So let's see what happens to the cell studies. We treat the cells, this is also Hela cells, with methylene blue and dimethylene blue. And here the curves in the dark and under illumination. And here we have micromolar concentration, okay? The IC50 is around 10 micromolar. And here we're doing the same assay with dimethylene blue, but the concentration here is 100 of nanomolar. So if you calculate IC50 for dimethylene blue, this is keratinocytes, another cell type, the result's not so different. The IC50 for dimethylene blue is 10 nanomolar, and for methylene blue is 2 micromolar. So this is 200 times more efficient. And why is that? That's what we want to answer. And the other things we see also is that when we treat the cells with methylene blue, with this is a dye that's, that targets acidic organelles, we see that it has lysosomes, and when it's not targeting the organelle, it has a green marker. So that's a normal wave. It's very similar to the control. Control, 4 micromolar methylene blue, and when you do this with dimethylene blue, we see a lot of lysosome accumulation. This dye accumulates much more efficient in lysosome. So there is a first evidence that somehow lysosome is involved on this. So can we measure reactive oxygen species? We do the same we did in the other result I showed to you. There is a relationship here between increasing concentration until you reach the IC50 for methylene blue. There is an increase in reactive oxygen species generated inside the cell. And for dimethylene blue, you don't see anything actually. You can see a very little increase. So you kill the cell, but you don't see these reactive oxygen species. And the reason is because you're generating it in the right place to do the specific task. So if we look a little bit more into details of the data I showed to you, here is the dimethylene blue with acridine orange. You see there is a lot of lysosome marker, similar to what we see in chloroquine. And chloroquine is an inductor of autophagy. And so how can we make sure really this process, where is this going? Is this really happening here, the accumulation of this, or is it accumulating here? So we have to do some more tests. I'll just show you one example of the things we are doing. We are treating the cells with several markers at the same time. We mark the nuclei with this DAPI. We mark autophagosome with a protein that's called LC3. We mark lysosome with another specific protein that's called LAMP2A. And if there is colocalization, we know that the process is going up to here. And here is the control and the methylene blue. We don't see any marker of this or that because we're not affecting in any meaning the autophagic flow. But when we treat with chloroquine or dimethylene blue, we see this colocalization. It means that we are killing the cell by autophagy. So we are now in the process to try to understand all these details. Svacha, Dr. Svacha is here. He's the guy that's trying to finish these experiments to make sure, is it the mitochondrial mitophagy involved? How much is involved? And so on. But we are sure that we can increase the efficiency of PDT hundreds of times if we induce autophagy instead of just killing the cell by making a lot of reactive oxygen species. So now I'm going to change a little bit gears here, and I'm going to show you some data that is the work of Patricia Santos. She's a student that was supposed to be here, but she had no more fellowship. She got a job. She cannot come anymore, but I'm presenting her data. The poster, the abstract is in the book. You can read. The thesis she defended is the following. if you look into the main problem of oncology, you talk to any doctor that works with oncology, and he asks you, is this technology that you're talking about going to resolve metastasis? The problem of the cell going from one initial tissue to the other. And that's the question. And I think PDT has not been able to solve this so far. So we ask ourselves, can we do something about it? Can we prove that PDT can facilitate or make it more difficult for the tumor to do metastasis? So when we think of metastasis, we think of a carcinoma becoming invasive. So the cell initially has to detach from this, but it has to do a lot of migration that involve adhesion and detachment. And when it reaches a new tissue, it has to face resistance to anoics. Anoics is a kind of cell death, apoptotic cell death, that happens when the cell detaches from the environment it normally has. And when it forms a new tissue, it needs a lot of adhesion. So if you look here, adhesion is a very important issue in terms of making a new metastatic tissue. And then what Patricia asked herself is PDT affecting adhesion somehow? So that's the question of the thesis. So is there any background history on this in PDT? There is. So the several studies we found in the literature conclude that PDT actually increase the adhesion efficiency of cells to substrates. And the main mechanism of doing this is cells avoiding trypsinization. So you put the cell in the culture, you treat with trypsin, that's a protein enzyme that cuts the matrix protein, so release the cell. If the cell has resistant to trypsinization, it doesn't get to the solution. There is some adhesion assays in which you measure actually the cells, how strong it's adhered, and now you see results with both increase and decrease adhesion, the effect of PDT. And when the people in this literature, especially this one, saw that there was an increase of adhesion, he concluded that there is some other mechanism involved because there should be increase of adhesion instead of decrease. Anyhow, there is not conclusive experiments, and we need to use more elaborate techniques because in these ones you can actually measure kinetics of adhesion. You just measure final product. You may get in trouble with that. So what Patricia did initially was to develop a technique in which she can really quantify adhesion as a function of time. And this technique, this is one of the techniques she can use, and she developed this. It's called reflection interference contrast microscopy. So it's basically an interference methodology in which you can measure the refractive index that's very close to the surface. And the reason is because when you have interference, you have positive and negative interference, and when you have the cell adhere to the surface, you have negative interference, you see black. So she can just measure the amount of black and white in the cell and can measure area of adhesion. And so we did these studies by using several molecules. I mentioned here three of them that are more important. One is PP9, and then Patricia and a colleague, Professor Shaw, devised a way to connect PP9 to polylysine. Very simple chemistry here. They saw that this is a polymer in which you have several porphyrin units on it. And there's TMPP, that's also a molecule that's well charged and used for PDT studies. And so what she saw when she started doing experiments in the dark is that incubating the cells with the photosensitizers in the dark would increase a lot adhesion to the surface. And this was not true for this PP9 or for the polylysine one, but for any molecule she studied, all of them increase adhesion. So it's very likely that the previous result was not because of the PDT effect, but because of the dark effect of photosensitizers interacting with cells. And we know that at this short incubation time, the molecules are basically in the membrane. And when they are in the membrane, they change the membrane properties, especially the fluidity of the membrane. And the other thing we saw is if the molecule is amphiphilic, if it binds more efficiently to the membrane, it increases further the adhesion area. So what Patricia proposed in her thesis is the following. When there is intercalation of photosensitizers in the membrane, you have the larger the membrane interaction, the larger the membrane area. If the lipids get area to expand, they get more flexible. And if they get more flexible, they can deal better with this adhesion force that are basically two. One of the force is to avoid adhesion. That's because of the glycocalyx of the cells. They hinder adhesion because they are bulky molecules and it's difficult to get together, and the specific interactions. If the membrane is flexible, this is easy and adhesion is stronger. So what happens after irradiation? What we see is that this molecule doesn't do much after irradiation, but this polymeric porphyrin totally inhibits adhesion. And so we want to study more of this. Why is this happening, molecule being able to modulate cell adhesion? But the fact that we show that in both cases, for porphyrin and for polylysine, both of them, when there is PDT, there is a decrease in adhesion. So the efficiency of the cell to adhere to substrates is decreased if there is photooxidation reactions. So I think this is a good thing for PDT, because if the cell cannot adhere to new substrates, it's likely to have a smaller efficiency to get metastasis. But this is for you people to prove. I don't do clinical studies, but that may be one thing to test. So why is this cell completely hindering cell adhesion? We have now data showing that it's because it's inducing cell apoptosis. The cell membrane is still not allowing incorporation of iodide or preputium iodide. So the cell is not dead by necrosis, but we have data now that I don't want to show because this is initial data, but I can talk to you about later, that is happening, the cell is getting killed by apoptosis. So we didn't explain to you yet why is this molecule more efficient than this one to induce PGT effect. And so I want to go a little bit quicker here to show to you some experiments we did with proteins. Okay, matrix proteins that are covering the cell in the tissue is PGT. If we photolyze these proteins for fetal bovine seroalbumin, if we're photolyzing it, is it increasing or decreasing adhesion? And then what we see is that this is the control adhesion of the control cell. When you treat the serum that is used to treat the cells, we didn't treat the cells in these experiments, just the serum that has proteins. Nothing happens if this is a porphyrin, but if we treat the serum with the polylysine porphyrin, the cell cannot adhere. So in the same way, this molecule seems to be more efficient, and this result depends on the amount of reactive oxygen species you generate. If you put less of that, if you put a reducing agent, you recover the adhesion. And so the first thing you may think of is the reason because this molecule is more efficient is because it makes more single oxygen. And then you have to test, and you put these two molecules in ethanol, and you measure single oxygen, and it's the same yield, the same lifetime. So it's not because one is more efficient in terms of photophysics than the other. It's because of something else. Who can tell me the reason why one molecule is the hypothesis? Can somebody make a hypothesis here? Does anybody? Huh? The reactive oxygen species here is what?
Yeah, the reactive, the generating species are more localized.
What do you mean by localized? I mean, it may be a.
They're all in the same area, so you damage the
Me-
same.
Oh, when it's in the membrane or in the protein.
Yeah, when it's on the membrane.
Well, that's an interesting aspect. We didn't test that. It may be true, but I don't know how. I don't have the answer to tell if it's true or not. It may happen.
Okay.
We didn't test that. No. There is other hypothesis that I tested. That it's not this one. That it proved why this is more efficient than other. But I cannot tell that this hypothesis that you made is wrong. We have to test that.
Okay.
You can come in the lab if you want. Well, what we did was we studied how is the efficiency of this molecule compared with this other one to photolyze proteins of the serum, okay? And this molecule, the polylysine, is more efficient, not so much, but it is more efficient to oxidize proteins and make carbonylated proteins. But still we don't know why. Why is this making more of this than this one? So we did some photophysical experiments, fluorescence, using a model protein that has a lot in the serum. That's a BSA, bovine serum albumin. And what we saw is that if you put a PP9 in the presence of bovine serum albumin, there is a big shift in emission. And the reason is because at this condition, PP9 is dimerized. And when you put BSA, BSA has hydrophobic sites. And the emission is shifted. If you do the same experiment with this molecule, nothing happens. There is no shift in maximum. If you put 10 times more BSA than what is in the serum, you do see a little bit of decrease in fluorescence. So you think it's binding somehow to this protein, but superficially. And it's still leaving these molecules open to react with oxygen and with other targets. Can we prove that? We tried. Okay, if you take PP9 and you measure single oxygen, in the presence of BSA or without BSA, there is a large decrease in the presence of BSA. And the reason is because when these molecules bond here inside the hydrophobic site of the protein, there is no way the oxygen can reach the molecule. Can we target the triplet species also? And we did the experiments with the triplet species. This is the triplet of the polylysine. If you put BSA with or without BSA, the lifetime is similar, around 500 nanoseconds. The lifetime of this molecule is in the nanosecond time scale because it reacts with oxygen and forms single oxygen. If you put PP9 and try to make the triplet, you don't see anything in solution because it's dimerized. But if you put in BSA, you see a triplet, but the lifetime is in the microsecond. So it's hidden inside the protein, it doesn't react with oxygen. So by adding this molecule to the polymer, we were able to tailor its properties in order to allow it to still react with form single oxygen, even in the presence of proteins. I think, in my opinion, we have not done these experiments so deep in the cell yet, but I think this also explains why the molecule is more efficient in the cell studies. Okay, so I want to shift another gear, and I just want to mention one date. I'm not going to further details on this. Some time ago, 2 years ago, we were able to show that if you excite melanin with the visible light, you see emission of single oxygen, okay? And you can do that with any melanin you think. I got melanin from Japan, from the US, I made melanin in my lab, I extracted melanin from here. Any melanin I use, I see a spectra. It varies, okay? If you look in this date in this paper, there is more details of this. If you treat the melanin with heat or if hydrolysis with urea of peroxidation, you open the structure of the melanin and it makes more single oxygen. And besides producing single oxygen, melanin also reacts with single oxygen because it decreases its lifetime, forming a hydroperoxide on the disposition here. So I want to ask a question for you now. Why is this important for you? Why do you think this is important? She wants to say something. You don't want to say anything? You were. Please say it to me. Yeah. Tell me.
It's important for melanoma.
Well, that's true. Why?
Because it would be a target.
Well, let's
the
think of-
metastatic cells has a lot of melanin.
Okay. Let's think of you then. Do you go to the beach? Do you do sunscreen? You put a lot of sunscreen. You protect against UVA and UVB.
And visible light.
Do you protect against visible light?
Yes.
You do?
Yes.
How?
Because the new sunscreens, I am a dermatologist.
sorry.
Of course, I protect of visible light.
Okay.
Some new sunscreens, they said to me,
Okay.
because the new sunscreen has some physical filters.
Okay.
And they are able to protect from visible
Yeah,
light.
I think this is the main thing I want to mention to you. I mean, some people know this, some people don't know. This is happening now, similar way that happened with UVA 30 years ago. When I went to the beach, I barely protect against UVB. But I left UVA on me all the time. And people from Photobiology Society already knew that UVA caused damage in DNA. UVA caused several problems, but normal people didn't know. And still, I think the definition of how to protect against UVA is 2 years old in Brazil, right? I don't know if Anvisa has already decided how to do the test, but if it decides, it's 1 year old. So still, we are learning to protect against UVA. But you have to know that if you go to the beach and you protect against UVA and UVB, you still are getting a little bit of burn with visible. So we have some data showing that this is damaging the hair. Of course, there's a lot of melanin here. And it's damaging skin cells. If the cell has more melanin, it makes more single oxygen. And if it makes more single oxygen, it dies more, has more phototoxicity. My student has done this tunnel assay. I'm not presenting here. He has to repeat it. But he has very clear evidence that visible light also damages DNA if you have cells with a lot of melanin. So we have to be careful. I want to finish by acknowledging lots of people. These people that have 3 stars are the ones that contributed to the work I showed to you today. So the green ones are people that are working in my lab still. The blue ones, like Adil Shoa, are participating somehow. The black ones are people that collaborate, Professor Oliveira, and the people also. Professor Tardivo, he'll be here tomorrow. Talk to him. Very nice lecture. The experiments with the adhesion was done in collaboration with people in France too. The group that's called M-Cube. They do a lot of studies with the membranes. And I want to thank you, and I hope I can answer any question.
Thank you, Professor. Questions?
Hi, thanks for your presentation. You answered some question that I had before. That I have a question. Did you try with some molecule with some metal ion, like copper, for example, or another metal ion?
Yeah. There are lots of studies with porphyrins that have metal ions inside. Is that what you mentioned?
But a small molecule, because it reported that a small molecule than porphyrins that has anti-tumoral activity per se without any light. I don't know.
So you're talking about cytotoxicity in the dark?
Yes. And maybe will improve with light and can you read?
Yeah. Well,
because you are talking about selectivity.
Yeah,
in
yeah.
this molecule?
I agree that you may improve a lot if you do symbiotic mixtures with two molecules, one do something, the other do some other things. There are some studies in the literature. I don't know. I don't do that, but I can give you some reports on that. But I don't know if that's what you meant. Symbiotic studies. You put a molecule that works in a certain cell target in the dark together with a photosensitizer and do PDT. Is that what you're saying or not?
Yes.
Yeah, there are some people doing that. I mean, I want to mention something. There is a method called PCI, that's called photochemical internalization. That some people in Europe, they have devised it now, doing some clinical trials. And they were able to show that if you put a photosensitizer that damage lysosomes, it's phthalocyanine asymmetric, negatively charged, the most one used. You increase a lot incorporation of chemotherapeutics, drugs that are used in chemotherapy. And so they are seeing patients improve a lot if they do PCI together with the chemotherapy. I think it's in that direction, right?
Yeah.
I can give you some literature.
Okay, thank you very much.
Yeah, I have a couple of questions. The first one deals with the is the path that you use to control whether it goes through necrosis or apoptosis. Is that specific to cell lines, or is that any cell line will undergo?
We have tested some, and the process I mentioned here is working all of the tested ones, because the molecule itself has a high tendency to accumulate in the membrane of any cell. And the other molecule that's inside the nanoparticles can only enter cells by endocytosis. So it doesn't accumulate in the membrane. This process is working for all the cells we tested.
Okay. And then, my other question was for the when you generate the reactive melanin derivatives, what's the time frame that it takes for the visible light to actually generate those species?
Okay. The generation is very quick. We have data showing that if you take a hair and you measure the single oxygen lifetime, it's much shorter than it would need for the single oxygen to leave a hair fiber. So it's generated and it gets killed inside the hair fiber. I want also to mention something that I didn't mention in the lecture. Melanin is very, very important to protect against UVB. Okay, that's people that have more melanin in the skin are much more protected against UVB. That's for sure. Not everybody knows that. But even people that have more melanin, in terms of visible light, they may have more effect because the protection is not working there. But to answer your question in terms of the lifetime, so melanin generates not a lot. It's a very small amount, but it does generate single oxygen. But it also reacts with it. So in the end, what you do, you kind of damage your own melanin. We see the hair gets photobleached. In the cell, we see also losing color. But it's getting killed in the cell. The hair doesn't get killed, but it loses color, it loses other properties. So I don't know if I answered all your question totally. If you have any more, please do ask more questions.
Hi. Thank you for your presentation, and I'm curious about melanin because I was thinking, if I expose myself to some light, you produce more melanin to protect yourself. So,
yes.
You said that this can produce oxygen. No, so I'm sorry. Yes. So, okay, this is toxic or not, or just with this specific wavelength?
Yeah,
I'm not confused, and it's a paradox for me.
I forgot to do something I always do, and because of the time, I didn't do it this time. Melanin is very important to protect against UVB, okay? So if you have damaged your cells in the basal membrane, the only mechanism it has to protect themselves is to produce melanin, okay?
Okay.
If you have more melanin in your skin, is your better protection against UVB and also against UVA. The problem is nothing in nature is 100%, okay? If you take any plant, actually, take any plant, it's there living from the sun, right? You think it's not making single oxygen? It is. And that's why every 2 hours they have to make a new reaction center.
And you have enzyme to protect it, of these species.
So if you stay in the sun, you think you're protected, and you're not totally protected. That's the main thing. You should avoid staying too much in the sun, because even the visible is causing damage. And we know there are some molecules that do photochemistry in the visible. Melanin is not the only one. Flavins also do that, okay? Flavins also absorb in the visible. They also do triplets. They also do single oxygen. The thing why I mentioned melanin is because something we discovered some time ago that makes single oxygen. Some other people have seen before that it makes other reactive oxygen species in the UV. So it's not only single oxygen. Melanin is protecting you, yes, but it's not 100%. If you expose yourself, even in the UV, melanin, if you have more melanin, you'll be producing more, but melanin is also protecting, it's also an antioxidant. So there is a balance that every person has its own. And you should know that. That's the thing I want to say. I don't know if I was clear. If I'm not, I can try again.
Yes, thank you. It's okay.
Mauricio, thanks a lot for your very nice talk. It was very inspiring for us, mainly in terms of organic synthesis, because your molecules is very beautiful and your work is very beautiful too. I have a question about the phenothiazine derivatives. Is it possible to use, for example, methylene blue in vivo treatments? For example, can you inject the methylene blues in your veins?
Yes, I mean, this is used for newborns. Okay. If they are born with a disease that's called methemoglobinemia, hemoglobin with the iron plus 3 species that doesn't bind oxygen, if this newborn will die. And what they do, the physicians inject 2% methylene blue intravenous. So the newborn will, how do I say?
Eliminate?
Urinate
Yes.
blue for some time.
Okay.
And so it's a very safe molecule. That's the main reason why most of the studies Tardivo does is with methylene blue, because it's safe.
But it's only in topic PDT, okay?
I mean, he has injected, but it is a new protocol, but it's safe. I mean, it's used for some disease, especially the one I mentioned to you.
It's because I've heard that methylene blue, for example, is very toxic for using in internal PDT. But I checked that you use it in small concentrations, for example, in nanomolar concentrations.
No, this is dimethylene blue.
Dimethylene blues, only in nanomolar concentration.
Yeah. Methylene blue has to be 1%.
Okay.
But it's, I mean, it is a safe molecule. You can check that.
Okay, thank you.
More questions? No? So let's thank him again, Professor Mauricio.